Hi everyone, in this lecture we continue exploring the development of Drosophila, which we began by covering some aspects in the previous lecture. And I want to remind you that understanding the development of this organism is important because Drosophila is a useful model that has been invaluable to our understanding of development and disease. In fact, this organism has even been used to study attention deficit disorder. In the previous lecture, we discussed the distribution and the effect of the maternal effect genes, and these are the first set of genes responsible for specifying the anterior-posterior axis in Drosophila. And these are known as maternal effect genes because they are deposited as mRNAs prior to fertilization. These genes are responsible for the specification stage of cell fate commitment in Drosophila. And recall that specification refers to the first stage of commitment where the cell fate can still be altered. The second stage of cell fate commitment determination, at which cell fate is no longer flexible, is mediated by a large group of genes collectively known as the segmentation genes, and those include the gap genes, the pair-rule genes, and the segment polarity genes. Before we discuss the different segmentation genes, we need to first understand what is meant by segment and parasegment in Drosophila. The epidermis and mussel layer of Drosophila has distinct segments, each of which has a unique identity. For example, thoracic segment 2 has both legs and wings. So each of the segments that we see in the larva correspond to a segment in the adult. And recall that these segments become apparent during gastrulation. What has been observed is that the expression patterns of some embryonic genes and the arrangement of some adult tissues, like the nervous system, don't quite correlate with the segments that we see in the epidermis and mussel in the larva and the adult. They have instead what is known as a parasegmental organization. A parasegment is composed of cells from the posterior compartment of one segment and the anterior compartment of the next or more posterior segment. As you can see, some genes like FTC are expressed in alternating parasegments rather than in alternating segments. So the parasegment appears to be the unit of embryonic gene expression. In the case of the nervous system, having a motosegmentation that is different from that of the muscles may be important for helping to coordinate movement, because one nerve can coordinate the movement of two different segments. Of the segmentation genes, the first set of zygotic genes to be expressed are the gap genes, and the gap genes are known as gap genes because gap mutants lack large portions of the body extending several segments. The reason for this is that gap genes are expressed in one or two broad regions of the body, and those regions are composed of multiple segments. The next set of segmentation genes to be expressed are the pair-rule genes, and pair-rule gene mutants are missing portions of every other segment. The reason for this, as we'll see, is because these genes are expressed in parasegments, which, as I mentioned previously, are composed of the posterior region or compartment of one segment and the anterior compartment of the next segment. Finally, the segment polarity mutants have different types of defects in every segment. This could be an inversion in the polarity, a duplication, or a deletion of the segment.